Hello guys, welcome to my YouTube channel of physiology. Today our topic will be neuronal physiology. In neuronal physiology, we will be discussing about graded potentials, short distance, short range electrical signals. If you have a good idea about membrane potential and how membrane potential is established, then things like graded potentials become much easier. And I have already uploaded topic on membrane potential on my channel. You can watch it. In graded potentials, we will be discussing its ionic mechanisms and movement of ions across the cell membrane and along the cell membrane during graded potentials generation and their propagation. Before proceeding any further, we must have an idea about the structure of a neuron. A nerve cell or neuron consists of an expanded portion, that is cell body, and a single long branch is axon. The junction of axon with cell body is axon hillock. It is trigger zone, and axon itself is conducting zone. At the end of axon there can be various branches called as axonal terminals. They are output zones. At the end of axonal terminals there are round structures that are synaptic knobs or terminal buttons. Inside the synaptic knobs are membrane bound vesicles having neurotransmitters as shown in diagram. Axon may be covered with sheet of lipid called as myelin sheath. When axons are myelinated, they will have myelin sheath, and when unmyelinated axons are in neurons, then they are called as unmyelinated neurons. They do not have myelin sheath. In central nervous system, this lipid covering is formed by oligodendrocytes, and in peripheral nervous system this myelin sheath, this sheath of lipid, is secreted by Schwann cells, and these little branches around the axon are dendrites. These are the input zones. Now, in neuronal physiology, we will be discussing about electrical signals or electrical current, messages in the form of electrical current that are generated due to the changes in resting membrane potential. And we know the magnitude of resting membrane potential is minus 70 millivolt. If changes in resting membrane potential are in small range, that is 15 to 20 millivolts, for example, resting membrane potential changes from minus 70 to minus 50 millivolt, then the potential difference that is developed, it would be 20 millivolts. And if resting membrane potential changes from minus 70 to minus 55, then the change in voltage would be 15 millivolt. So these small changes in resting membrane potential are called as graded potentials. Graded potentials are small distance electrical signals because they can only travel 1 to 2 millimeters on axon, and their propagation is decremental as they move away from the region of their origin. The magnitude of change decreases. That if 20 millimeter of change has occurred during its propagation, it will gradually start to decrease. Therefore they are called as small changes in resting membrane potentials, and they serve as small distance electrical signals. Large changes would be discussed in the topic of action potential. Now, let's come back to our neuronal physiology introduction. Neurons and muscle cells are excitable cells. And excitable cells and tissues are those cells and tissues that undergo changes in their membrane potentials, and these changes are called as electrical signals. Electrical signals are important for communication between the cells and coordination between the cells, release of chemicals from the cells. For example, a cell has to release neurotransmitter or a hormone, then there must be some sort of electrical signal that stimulates it to do so. Similarly, skeletal muscle cells contract and relax due to electrical signals. So the change in resting membrane potential is electrical signal. Resting membrane potential of a neuron is minus 70 millivolts. In previous lecture, we have discussed resting membrane potential is established due to uneven distribution of charges in extracellular intracellular fluid, and these charges are unbalanced, and also due to the difference in permeability of ions. For example, cell membrane of a nerve cell is 50 to 75 times more permeable for potassium ions as compared to sodium. And similarly, trapping of anions like proteins inside the cell, that is in intracellular fluid, also contributes to the internal negativity of a cell. And sodium potassium ATPase pump also contributes to the establishment of resting membrane potential. We have revised all these concepts again over here because change in resting membrane potential serve as electrical signals. And still there are some other definitions like polarization. Polarization mean unequal opposite charges separation. If a cell membrane have 0 millivolts, it means it is not polarized. Fortunately, scientists have long axons that can be seen with naked eye, and they have isolated them from mollusk like squids. If microelectrodes are placed inside and on outside of a axon, in the axon of a squid, it will show the voltage. When a voltage value is zero, it means there is no polarity, there is no separation of unequal opposite charges. Now, greater is the number of opposite charges separated, greater is the potential. Now consider case A, B, and C. In case A, only three opposite charges are separated. Here there are four charges are separated, and here this is the greatest number of charges that are separated over here. So this membrane is much more polarized than this, and this membrane is more polarized than this one is the least polarized. Depolarization mean any decrease in internal negative polarity, any decrease in internal negativity, that is change from minus 70 to minus 60, is depolarization, decrease in internal negative polarity. Any decrease in resting membrane potential is actually depolarization. Repolarization is opposite of depolarization. For example, a membrane changes from minus 50 to minus 70, it is repolarization. When internal negative polarity is regained again, it is called as repolarization. Any increase in internal negativity from resting membrane potential would be hyperpolarization. Resting membrane potential is minus 70 millivolts. If internal negativity increases further away from minus 70, it would be hyperpolarization. Changes in membrane potential or resting membrane potential serve as electrical signals. In this case, if membrane has changed from this value to this value, this change in membrane potential would give rise to electrical signals. As I have already discussed this, changes in membrane potentials are electrical signals. If they are small changes in there are small changes in resting membrane potential, they are called as graded potentials. And if the changes in resting membrane potentials are large, for example, resting membrane potential from minus 70 millivolt changes to plus 30 millivolts, now the change is 100 millivolts, then it would be called as action potential or large distance electrical signals. We will be discussing this in next lecture. Now we will only concentrate on graded potentials or small distance electrical signals. Now graded potentials, some sort of triggering event. It can be electrical signal, chemical signal, or mechanical signal. It is gonna to open the gated sodium channels. As in extracellular fluid, concentration of sodium is much greater than intracellular fluid. Both electrical gradient, because outside is positive and inside is negative, electrical gradient favors the sodium movement from outside to inside. Similar concentration gradient also favors sodium movement to inside. Any one of these signals can open this gated channel. This is the closed sodium gated channel. Now this is the open sodium gated channel. Now we are not going to discuss what is the triggering event. We have discussed it can be among, it can be one of the signal. It can be electrical signal, it can be chemical signal, it can be mechanical signal that is gonna to open these sodium channels. When sodium moves from outside to inside, from extracellular to intracellular fluid, these blue colored circles having positive charges means sodiums that have moved in. So this is an active area. It is more positive as compared to this adjacent inactive area. And there is a decrease of positive charges due to the movement of these positive charges to the inside. Now, these positive charges from their higher concentration can move towards this area, and on inside, these positive charges can move from more positive to less positive. That is, they can move from positive to negative area. On outside and on inside, charges will be moving in opposite direction, and same would be the case over here. So this movement of charges, that is between active area and adjacent inactive area. Adjacent active area is still at resting membrane potential. Now their change has occurred due to the movement of positive charges from outside to inside. And this movement across the membrane is electronic conduction. And this conduction along the cell membrane is passive conduction. And this would be our graded potential and small range electrical signals. When sodium has moved from outside to inside, the small change in potential that has occurred, it has given rise to these small range electrical signals, because charges move from active area towards the adjacent inactive area. So the net flow of charge is electrical current, because when uh, sodium has moved towards inside, charges start to flow from active area towards inactive area. So this is the net flow of charges, that is current. And this current act as short range electrical signals, small distance electrical signals, and small changes in membrane potentials are graded potentials that have given rise to this electrical current. In this specific case, charges move from active area to adjacent inactive areas. As I have already told you that triggering event can be chemical, electrical, or mechanical stimulus. And correspondingly, if stimulus is chemical and channel would be chemically gated channel. If stimulus is electrical, then channel will be voltage gated channel. And if stimulus is mechanical, then gated channel would be mechanically gated channel. Graded potentials are decremental. As positive charges have moved from extracellular fluid towards the intracellular fluid, due to this movement, due to this electronic conduction, the membrane potential has slightly shifted from more electronegative towards less electronegative, and this graded potential has given rise to the flow of positive charges both on outside as well as on inside. This is passive conduction. These are the short range electrical signals. But when these positive charges are leaked through the open channels, leak channels of sodium, then again internal negativity would be restored. And when there would be no membrane potential, then there would be no conduction of electrical current along the length of axon. Simply graded potentials die off. They are decremental, they gradually decrease. Because potentials and electrical current are related with each other by a relation called as Ohm's law. When there is no voltage, there would be no current. So when magnitude of graded potential is decreased while traveling due to the leakage of sodium ions, the current that is traveling would also be less. Now, finally, graded potentials are effective only for small distances, that is from 1 to 2 millimeters. Graded potential spread is called as passive conduction, and loss of current across the cell membrane. When sodium from inside to the outside moves through the leak channels, then it would be electronic conduction. Electronic conduction or local current loss is responsible for gradual decrease of graded potentials. And one thing that I want to share with you at the end is greater is the magnitude of graded potential, greater would be the electrical current, and greater is the strength of triggering event, greater would be the graded potential, and greater is the duration of the triggering event, greater would be the duration of graded potential. Next lecture we will be discussing about action potentials. Thanks for watching.